Hello everyone, today I'm here to present my project on the topic finding DNA promoter regions with the help of deep learning techniques. So we know that DNA promoters are crucial regulatory switches that control gene expression, but they are super hard to identify computationally. If you take the case of humans, about 24% of human promoters contain Tata box motifs. The majority lack this, making conventional motive scanning tools unreliable. This is especially problematic when it comes to studying about diseases that is linked to gene mutation, etc. Um, the novelty that we bring is, or the solution that we try to bring, is to combine two powerful neural networks, namely CNNs and RNNs. So the CNN is used to detect local patterns like data boxes and bi-LSTMs or the RNN that analyze long-range sequence context. To reduce false positives, we have implemented one more thing, which is to generate hard negatives, that is sequences that look like promoters but they actually aren't, by strategically randomizing only 12 out of 30 segments of real promoters. This preserves biological realism while training the model to spot subtle differences. Promoters are short DNA sequences, um, around 300 base pairs long, that act like switches to turn genes on or off. They work by attracting RNA polymerase and transcription factors, which are necessary to start the process of gene expression. Now, why exactly is it necessary to detect them? It is very important because it helps us understand the root cause of diseases like cancer and genetic disorders. And it's also essential in synthetic biology where scientists design and build gene circuits. Now, there are two types of promoters. About 24% are Tata promoters, which are easy to find because they contain a specific sequence like Tata box. But the remaining 76% are non-Tata promoters. These don't have any fixed or conserved patterns, so they are much harder to detect. Current tools like CNN-PROM or DNA-BIRD do well with Tata promoters, but they often miss known Tata ones, which means many true promoter sequences go undetected. Now, our solution is that we are about to build a hybrid CNN RNN model. So the CNN model finds the motifs and the bi-LSTM, which understands the sequence context. It's trained on humans, mouse, and housefly genes as well. Now, coming to why exactly does it matter? Because it gives an accuracy of 98.2%, much better than the older tools, and it also helps in detecting known Tata promoters, which older models often used to miss. Coming to the objective of the project, our objective is to develop a hybrid model combining CNNs and bi-LSTMs to detect promoter regions in DNA. CNNs help detect local motifs while bi-LSTMs understand sequence context. We focus especially on non-Tata promoters which are harder to detect, aiming to reduce false negatives by 30% compared to old models like CNN-PROM. Finally, we want our model to perform well across several species, including humans, mouse, fruit fly, with consistent MCC above 95%. We benchmark our model against CNN from DNA BERT and D promoter, aiming to outperform them in precision and recall. The goal is to be top ranked in balanced prediction quality. Our model can help in disease research by accurately identifying promoter regions. We aim to discover at least three new promoter linked to cancer, enabling further biomedical insights. Promoters are short DNA sequences located upstream of genes. They help start transcription by binding RNA polymerase and transcription factors. Tata box promoters make up around 24% of human genes and have a clear Tata sequence. They are found about 25 to 30 bases before the transcription start site and are linked to stress response or tissue specific expressions. On the other hand, 76% are non-Tata promoters. They use other motifs like GC box or DE and usually regulate housekeeping genes, but they are harder to detect because they lack a clear sequence pattern. One major challenge is false positives. Random Tata-like sequences can appear without actually being functional. For non-Tata promoters, the diversity in motifs make detection harder, and promoter structures um, are also varying between species, adding more complexity. So the current tools that we have, like CNN Prom, works well for Tata promoters, but struggle with non-Tata ones. Uh, given given an MCC around 0.77, DNA BERT performs better overall, but is very resource heavy, especially when working with large genomes. Coming to the methods that we used a 300 base pair promoter sequence from the PDU database for humans, mice, plants, and fruit flies as positive samples. For negative samples, we used a mix of randomly mutated promoter sequences and random genomic regions. To keep training balanced, we used a 1:1 ratio of promoters to non-promoters. Now, coming to the model architecture, our model combines three 1D CNNs with different kernel sizes, 3, 5, and 7, to detect various motive patterns. Then an RNN model, in this case it is a bi-LSTM with 32 units, which captures the forward and backward context in the sequence. Finally, we use two dense layers ending with a sigmoid output to classify the input as promoter or non-promoter. The ones with probability greater than 0.5 will be considered as promoter and the other as not. To ensure that our model is robust, we use hard negative mining. This technique involves replacing 12 out of 20 segments in real promoter sequences, each of length 15 base pairs. The goal is to present the model with more challenging examples, helping it learn better to distinguish between positive and negative instances. In terms of ensemble learning, we implemented, we are about to implement ensemble learning by averaging the predictions of the top 5 models which are ranked based on the MCC score. This strategy helps improve accuracy by leveraging the strengths of multiple models and reducing the impact of individual model weakness. Now, when it comes to hyperparameters, we chose the Adam optimizer, which is known for its efficiency in training deep learning models. To prevent overfitting, we applied a dropout rate of zero point 0.3. That is, 30% of the neurons are randomly turned off during training period to make the accuracies better for the models to predict promoter regions well. Finally, we set the batch size to 32, which helps balance the computational efficiency and the model's ability to learn and generalize features. Coming to model-based architecture implemented, CNN by LSTM hybrid model, we combined CNNs to capture local sequence patterns and by LSTM to understand the sequence in both directions, improving context understanding. Parallel CNNs, we use three parallel CNN layers with kernel sizes 9, 15, and 20, 21 base pairs to capture patterns at different scales. Bidirectional LSTMs they have 32 units and process the sequence both forward and backward for better context comprehension. A dropout rate of 0.3 helps prevent overfitting by randomly ignoring 30% of neurons during training. As you can see, our model achieves 94 to 95% accuracy on both promoter types with especially strong recall for non-Tata promoters, the harder case. The tight training validation gap confirms reliability. Next, ensemble models will be implemented to make predictions even more um, robust before expanding to other species. Mm, Also, one important thing to note here is that high precision means few false alarms. High recall means we miss few real promoters. That's what we have here. And other than that, the 3% recall boost matters because most human promoters lack data boxes. So this is the entire code for the model that we have developed. This is for data boxes. Four epochs have been captured and the highest accuracy is given by the 14th epoch, and here is the graph of the same. The training loss decreases steadily here, indicating the model learns from training data. Simultaneously, the validation accuracy also generally increases, showing good generalization to unseen data. The initial rapid improvement in both metrics suggests quick learning. However, the validation accuracy plateaus and slightly fluctuates later, hinting at potential overfitting if training continues excessively. Overall, the model demonstrates effective learning and generalization within the observed 14 epochs, achieving a final validation accuracy of 0.965. No, final validation accuracy is 0.9437. Monitoring the validation accuracy trend is important to prevent overfitting in future training. These are the confusion metrics just for the same. Coming to non-Tata boxes, we can see that um, both Tata and non-Tata boxes are showing high accuracy. However, in this graph, our training loss is again consistently decreasing, becomes quite volatile after the initial improvement. After the initial improvement, suggesting that the model's performance on new data fluctuated significantly. Despite the decreasing training loss, the instability in validation accuracy might indicate that the model is, here we can see that it's continuously fluctuating. the model is starting to overfit or that the validation set has some inherent variability. So further investigation into this model behavior and potential regularization technique must be needed to achieve more stable and reliable generalization, which is the reason why we are doing ensemble learning later. So this is the confusion matrix for non-data box. So coming to the summary of whatever we've built so far, the entire model was built from the ground up. The CNN scans for the motifs like data boxes by LSTM. The RNN checks the surrounding context. Our hard negative generation carefully randomizes only parts of real promoters that makes the model more precise. And the visualization tools that we used helps us to understand that our model isn't overfitting like we expected. And furthermore, we'll have to implement ensemble learning on top of this to improve the model. Thank you.